Well, hello, everyone. We're just a little bit past the hour, so we'll go ahead and get started to respect everyone's time today. Thank you for joining and welcome to the Digital Medicine Society's September webinar, introducing BARDA's new program, ENACT, Early Notification to Act, Control, and Treat, and how the ENACT program uses wearables for early illness detection. We have a very special treat for us today. We have the director of the ENACT program, Shayla Selamovic, to talk to us about this great new program. Before we get started, of course, we do have to do a little bit of touch base today about some housekeeping. So of course, as with all DIME webinars, we are recording today's session live. So be sure that whenever we ask some fun questions at the end, that it will be the fun will be recorded so we can all live it again later. So the slides will also be available afterwards, and I know that there is some really exciting stuff in these slides, so you'll really want to dive in afterwards. And so the fun happens with these DIME events is the live session where we have the live Q&A portion. So if you're on the line today, do feel free to either raise your hand and I'll unmute you to ask your question live, or of course you can always type your question into the Q&A box and we will get to as many as we can today. So without further ado, let's get to the reason that you're all here today, and we'll go ahead and get started with our fantastic presentation. Shayla?
Thank you very much, Rachel and Jennifer, for inviting me to speak to the DIME community today. I really do appreciate it, and I am very excited to be able to share with you the current status of this program. So let me share my slides. So I hope that you can all see this, and I hope that this is the correct view.
Looks like perfect.
It is. So thank you. So again, thanks very much for having me today. I don't know how many of you know about BARDA and ENACT, so let me just take a moment to place this organization for you. So we are part of the Department of Health and Human Services within the Office of the Assistant Secretary for Preparedness or Response, or ASPR. And ASPR's mission is, of course, to save lives and protect Americans from 21st century health security threats. Now, BARDA stands for Biomedical Advanced Research and Development Authority, and we are not a standalone organization, but we are part of ASPR. And so our mission and our goal is to develop and make available medical countermeasures, and we do that through unique public-private partnerships. And obviously our goal is, a major goal is really for us to drive innovation. We don't do basic research. We don't do research just to do research. So we are very different in that context than, for example, NIH. But again, because we are developing medical countermeasures, our goal really is to drive innovation, to get those medical countermeasures to the patients and help save their lives as soon as possible. So I mentioned already that we're part of ASPR, and here are the 5 main divisions. And just looking at these divisions, I think gives you a good sense of what BARDA is really all about. So we are very much interested in threats to national health security, and that's why you see, for example, a division on influenza and emerging infectious diseases. There's also a division on CBRN, and my program is housed within the Division of Research, Innovation, and Ventures. And I think that title already tells you something about the way we approach work. So part of this DRIVE division is ENACT, which stands for Early Notification to Act, Control, and Treat. And I do want to give a shout out to all of the members of this fantastic team who are supporting all of the projects within the program. So what is really ENACT all about? So in the traditional healthcare paradigm, a person is not aware that they've been infected, but they really only know it or are aware of it once they develop symptoms. Let's say fever, a cough, might be a sneeze, body aches, etc. At that point, it's very difficult to differentiate the illness, and the patient can often be a transmission vector even before they start noticing those symptoms. So once they experience the symptoms, they need to make an appointment with a healthcare provider to get a diagnosis. That often involves various ways to just get an appointment or even necessitates a visit to the emergency room. And finally, after that, if a diagnosis is actually made, it's time for treatment. And that requires, again, going to the pharmacy, filling prescriptions, etc. And at this point, because it's now a little bit farther along in the disease progression, some treatments may not necessarily work as well as they would have had they been given to the patient, 3 days ago. And so there is clearly a major cost to the individual, which is, primarily just actually being sick and developing a greater, stronger symptoms over time. But there also is a major cost to the community, including delays that cause a burden to the healthcare system, obviously a loss of productivity, and certainly in case of infectious diseases, there is a risk of transmission and spread in the community because, again, it takes a while until the disease infection has been diagnosed. And so we at ENACT are very ambitious in trying to upend this entire paradigm. So we're all about, again, as the title says, early notification that enables us to act, control, and treat the disease. So we're interested in supporting pathogen-agnostic technologies that enable this earlier infection detection. And we do that through 2 pillars. In the one pillar, we're really all about empowering individuals to take early action on their health. And I'll explain a little bit what that entails. The other pillar is providing early and actionable information to medical care providers, for example, for patient triage, for example, helping a doctor establish a diagnosis sooner than is currently possible. And last but not least, the third pillar is really kind of a larger context issue where we're attempting to develop technologies that can follow the spread of an epidemic in near time. And you can all imagine that that's actually turning to be really prescient and very vital during the current pandemic situation. And so, what I'd like to leave you with on this slide are, again, the 3 terms: act, treat, and control. So ACT really is focused on empowering patients to providing presymptomatic information about an illness. TREAT is all about providing early diagnostics to medical care provider or early diagnostic technologies. And then control is about providing actionable information to public health organizations. And so another way to look at this is, how does this really apply to each of these pillars? Well, in the ACT pillar, you can imagine this information allowing a patient to ask themselves, well, should I go take a sick day and see a doctor today, based on whatever sensors they're wearing, whatever digital signal they're getting. In the treat pillar, the doctor might actually ask, does the patient have a viral and bacterial infection? I think we can all agree that knowledge, especially if it can be obtained within 5 minutes during the first doctor visit, right, or even over the phone through digital medicine technologies is really important. And then again, the questions that we're asking in the control pillar are, where is the next hotspot, let's say, of influenza or another epidemic? And how can a hospital or a pharmacy use this information to better and more efficiently allocate the resources? And so I'd like to just dig a little bit more into what kind of questions we ask here, because at the end of the day, this is really a data issue for the ENACT program. We obviously utilize a lot of wearable sensors to obtain physiological information, biomarker information, general information about the health of an individual, right? So we are interested in developing various sensors, wearable sensors, activity trackers, etc. But at the core of the program, this is really a question, all pillars really contain questions about data analysis, right? So what do we do with all of these huge streams of data? Let's say if a person is wearing an activity tracker and the activity tracker, collects data for 24 hours and then for weeks on end. How do we analyze the data and what are we really trying to get out of it? And so in this, again, ACT pillar or pre-symptomatic information gathering, we're really analyzing data to attempt to find out whether there is such a thing as a health signature for an individual or a cross-population group. And I think general knowledge of medicine tells us, yes, there is something very general that should look like a health signature. I mean, we all know what a normal heart rate for a person of a certain age looks like, or a normal blood pressure range, etc. But, beyond that, obviously each person is different, right? And so we are attempting to detect, even small aberrations from that essentially normal signature. And so that's why I think it really is important for us to devote some effort to this question and say, well, does when this person is healthy, does the signature look the same every day? Or how does it change? And again, I think it's important to look at it across different population groups, by age, by biological sex, by ethnicity and race as well. And then, obviously the next question is, well, if a health signature exists, does a concrete illness signature exist for that individual or across population groups? In other words, I'm interested in finding out, if a person is, let's say, contracts influenza, does the physiological signature obtained from all of that wearables data always look the same whenever this person, gets influenza, or does it look differently every single time? And then, can we, just by looking at this wearables data by physiological markers like heart rate and saturated oxygen and blood pressure, et cetera, can we, just by looking at that, definitively determine what type of infection a person has? And again, it seems like we should be able to do that, but we're actually doing the really hard work of gathering all of the data, measuring all of these physiological markers, and then analyzing it. And, to some extent, I would say the perhaps most important question here is what exactly is the time period between the onset of this, I'm going to call it illness signature, and then obvious symptoms, right? Because that's the presymptomatic stage that we're interested in. If it turns out that for a particular disease or just in general, our sensors can detect this illness signature, 24 or 48 hours before the person develops, let's say, a fever or body aches, that's a game changer. If the wearables data tells us, well, we really can't detect much before maybe 4 hours, until 4 hours before, the onset of noticeable symptoms. Well, that's now a very different calculation, right? And so we're really, digging down here into the detail to definitively answer all of these questions. And then in the control component or pillar of the program, we're really interested in figuring out how early infection information can be useful to healthcare organizations. And so, if you're considering the current pandemic situation, right, the sooner we get data, or the sooner, let's say, a health department or a pharmacy or a hospital obtains information about a change in new diagnoses, be it influenza, be it COVID-19, be it some other infectious disease that might require hospitalization or treatment, right? The sooner they get that data, the better they can plan ahead. Right? If we see an uptick in influenza cases and that information is available on a day-by-day basis to local pharmacies, well, they know now they have to stock up on Tamiflu beyond what they had originally prepared for. And then last but not least, in the treat pillar, what we're really digging into is whether infectious disease can be detected or determined based on the host response and through on-demand tests. And so what I'd like to leave you here with is basically the image of, a patient that's visiting a doctor, and the doctor then administering on-demand tests that, are minimally invasive, perhaps only require a finger prick of blood, and that they can immediately tell, the doctor, well, we're dealing with a viral or bacterial infection, or whatever else it might be. And so I think it's important here to point out that we're not looking for an individual pathogen. Rather, we're looking at the host response to better understand how a patient actually responds to the pathogen, which I also think is really important because, for example, with COVID-19, we see that different people in ways that we don't fully yet understand have very different responses to this pathogen. And so just understanding which pathogen the person is exposed to is not sufficient. We really, I think, need to also look at the host response. And so, with that introduction, I do want to just give you an overview of our current investment areas. And again, these three investment areas, kind of this Venn diagram, I think also captures this vision of these three pillars. And what you see here is that actually currently most of our investments are in this pre-symptomatic empowering the patient field, but we are, expanding into these other areas as well. And I will briefly introduce some of some of our partners shortly. Because I'm talking here to the DIME community, I also wanted to just briefly touch on the variety of sensing mechanisms that we're supporting here, because I think it's really interesting and relevant perhaps to your community. And so, this showcases that we're using sensors that cover everything from electrodermal activity to ultrasonic detection to biochemical detection, accelerometry, pressure, etc. So it's a range of different sensing technologies. And this is, again, something that we're also interested in, to figure out which sensor is the best in whatever way, or most reliable, or, gives us the most succinct answers, perhaps. And then, similarly, we're also obviously looking at a host of different biophysical markers, just because it's not definitively clear which markers we actually need to observe to obtain that host response information, right, or that pre-symptomatic information. And so, of course, we've started with vital signs and monitoring activity, and we have several projects that are looking at blood chemistry, but we're also looking at neurological effects or neurological responses inside the body as a response to an infection. And last but not least, we are also, of course, including symptomology, but that's a self-reported biophysical marker, if you will, and so that's obviously a little bit less accurate than some of the other markers. But again, the point is here that we're really approaching this early health notification field with a very open mind and looking at a very wide range of biomarkers as well as sensor technologies, and then doing the hard work of data analysis to determine, what can we actually figure out from all of the data, which biophysical markers do we actually really need to monitor, and which sensor should we use. And so, because I think we do have some time, I do want to, briefly touch on actually all of our partners. And so I do want to also just specify that, I am covering all of the partners, and we don't have any preference towards one or the other. But again, because we are partnering with a range of different technological developers, I think it's really probably worthwhile to the DIME community here to hear a little bit about them. So the first subset, I think, of our technologies are biophysical continuously monitoring devices. And here we have Sonica, Spire, and UC San Diego. And you may have heard about Sonica. They've been highlighted a bunch lately in the press. Sonica, I think, really stands out because it's a wearable with a very interesting form factor. You can see here in the photograph on the left that it actually looks like a Band-Aid. It's a little bit thicker than a Band-Aid, but it is worn basically almost on the neck between the clavicles. And so it doesn't really interfere with, everyday activities. It is an acoustomechanic sensor. So it's not an acoustic sensor. It is an acoustomechanical sensor that can record information, for example, about the cough rate, and that can distinguish, different types of swallowing or, yelling versus talking at a kind of at a regular decibel level. And so it captures physiological signals that I think are very different from kind of the standard activity trackers, what we normally imagine when we talk about wearables. Spire Health also provides a very interesting form factor. So the health tag that you see here in the photo is actually attached to clothing. We conducted a study, a clinical study in partnership with Spire, where individuals actually wore these tags attached to their underwear. So again, it's supposed to be something that's very easy to implement in everyday life. Obviously, everybody's wearing clothes, so it's not something that you just forget to wear. And so this device was able to track the respiration rate, heart rate variability, stress levels, activity, sleep, and sleep quality as well. And then the next one is UC San Diego. This is a bit of an earlier project. In there, I mentioned that we're looking at neurological responses inside the body, and that's what UC San Diego is all about. So they're looking at vagus nerve firing to determine what the status of the immune system is, and they're hoping to be able to use these signals to distinguish between bacterial and viral infections. And so currently the form factor is a sensor or host of sensors rather that are worn on a band around the neck. Again, this is an early form factor. We'll see how this develops and changes over time. The other biophysical continuously monitoring devices are kind of the more, I think, more often seen wrist wearables. So we have here BioBeat and Empatica. BioBeat is actually an Israeli company and has this BioBeat watch that is already FDA cleared for blood pressure, pulse rate, blood oxygenation, 21 other parameters. And we actually partnered with BioBeat to use their device in a vaccine study and look at the presymptomatic response of the cohort to the influenza challenge that was applied during the study. Empatica is another partner. Empatica has this Embrace E4 device that we've been using that actually also is FDA cleared for seizure prediction. This device utilizes electrodermal activity to quantify the nervous system response. And one of the questions that we're attempting to answer with them is, could electrodermal activity essentially tell us something about the immune response? So next, we're looking here at a host of biochemical continuously monitoring devices. And for example, we've partnered with Enlicense to develop this sensor that's worn around the arm and that interrogates eccrine sweat and for the levels and the presence of multiple cytokines. And so, this is, again, an interesting form factor. The device can collect information for up to 72 hours before needing to be recharged or replaced. If anybody's interested in kind of a lab on a chip, MEMS types of those solutions, we've covered that as well. And so we're partnering with a lab at Stanford University to develop this microneedle patch that you can see here on the tip of the finger. It's really small, and again, it can quantify cytokines and small molecules in skin, essentially. And the goal is to develop it to the level where it can monitor multiple of these biomarkers for up to 2 weeks. And I think this type of technology is really also interesting, not only for pre-symptomatic monitoring, but also for drug dose management, right? Because it could potentially tell you how quickly a particular drug is being metabolized and, when the next dose is due. And again, this is a microneedle patch that's worn on the arm. We've seen that this type of patches is used for very different applications. There is, the nicotine patch, etc. For example, and people seem to be fairly accepting of that form factor. So that's one of the reasons why, we wanted to explore this microneedle patch and see basically, what the acceptance might be. Now, outside of kind of basic sensors, I should say, we're also investing in app development. And again, I've pointed out that at the core, ENACT is all about data analysis, right? So at the end of the day, all of these products development. And so that's also the case with our partner Evidation, which couples existing external fitness tracker data, so off-the-shelf devices that people already have at home and that measure, heart rate, ECG, steps, etc., information, and then takes that information and analyzes it through an app that they're developing. And so they're also collecting a daily symptoms questionnaire to be able to, again, correlate people's symptoms with the data from their wearables. They've also implemented an option to send the cohort members, I think it's over 5,000 study members actually at this point. So they also have implemented a way to provide to them a swab for influenza and a couple of other infections, a couple of other viruses that is delivered to the patient, to the user's home, and then can just be, mailed to the lab via the mail. And that step is used to provide essentially the ground truth, right? Does this person really have an infection? Do they really have influenza or not? But again, the core of this project is really developing this algorithm that analyzes all of this data, both the wearables data and the symptomology data, as well as then the ground truth, the PCR tests from people developing influenza symptoms. We also have one in-home monitoring solution by Sound Life Science. So they're developing a software that can transform commodity smart devices, for example, an Alexa, into an active sonar system. And so this particular device is stationary. It stays at home. You could envision it being placed in a person's bedroom so that they can monitor their breathing rate and respiration rate at night while they're sleeping. But of course, you could also imagine this actually being placed in hospital rooms of patients. Imagine a person didn't necessarily have to have, all sorts of physiological monitors attached to their fingers with all sorts of wires. It would be so nice if there was a device stationary in the room that could simply monitor from a distance of maybe a yard or so what's going on internally with that patient. And then we have a technology that's very different from all of this, which is really a telehealth solution. And that's in partnership with the company 98.6. So they've developed a telemedicine app that connects users to board-licensed physicians, right? So, envision a person developing symptoms, wanting to talk to a doctor without actually having to get into a car and drive there. They can obtain a diagnosis over the phone. The medical care provider can also order, let's say, an influenza test to be sent to the patient's home, etc. But a really interesting part here is the data analytics platform that allows the company to take all of that diagnostic data and provide a near real-time understanding of the disease spread by state and then nationwide. So currently, the company has over 500,000 daily users, and they're continuously growing. I think it's really interesting that they are able to provide, infection numbers, for example, for influenza, really in near real time, while it's, some other platforms, for example, CDC's ILI platform often has some delay of a couple of days and even up to 2 weeks. So again, this particular project that fits within our, control the infection pillar. And I believe this is the last set of projects. These are all early projects where we're really focused on point of care analysis. And I think these are really, very well placed for use in doctors' offices. So there's Rizlab Health that's developing a microfluidics-based blood counter that's then looking at the shape of different immune cells to basically determine what the host response is. Does the person have an infection, and if so, what type of infection? Is it viral or bacterial? Stanford Medicine, is developing a lab on a dust system that, again, on demand as opposed to the microneedle chip. This one can on demand quantify the multiple biomarkers in blood. The really great thing I think about this approach is that requires basically no prep. It's literally just a finger prick of blood and the answer is delivered within 15 minutes. So again, you can see, you know, how well suited this type of technology is for a doctor's office. And then, you know, last not least, we are also partnering with GE Healthcare to develop, to use ultrasound to detect changes in biomarker expression and immune cell responses in different tissues. So that is the gist of the program with a lot of detail. You know, I'm sure this was a lot of information. So if anybody here has follow-on questions, I'd be happy to address them now, or, you know, later via email. You can certainly reach out to us. Here are some websites that you can take a look at to see, you know, what BARDA is currently doing. beta.sam.gov contains our official funding announcements and information for all government contract solicitations. So if anybody's interested in what are we currently funding, which areas of interest are currently accessible and open and accept applications, that's where you would go. And then you can find out more at ASPR here and Drive as well. And I'm going to stop sharing my slides. And yeah.
Thank you so much for all that information. Fantastic stuff. Also love seeing unclassified slides. They are so exciting. You guys are doing some really excellent work. It's just seeing the reach of digital medicine just really evolve so much faster than even I can keep track of. It's just really exciting. It seems like the future of healthcare is here. And there is just so much really great work going on. So congratulations to you and to your team, and thanks for sharing all of this. So if you're on the line, I'm sure that you are doing two things, trying to digest the fantastic presentation from Shayla, and also thinking about what are the kinds of, feverishly trying to figure out how to go ahead and answer your questions. So feel free to raise your hand, and I'll unmute you to ask them, and you can also type them into the chat function. So while we are trying to gather our great questions, I do have some. And so you did share, and these slides will be available afterwards. And so, of course, take a look at them. But you did mention right before here about how to get in touch with you and ENACT. So I'd like to talk about that a little bit more in this conversation about, you know, so for a team to be on your radar, do they go to that website and fill out an application? Is that correct?
So we have our contact information published on our website, or I can, you know, type into the chat box the program resource inbox mail, so people can reach out to us. But yeah, I mean, essentially anybody can reach out to us. We actually talk a lot to both academics and then, you know, representatives from private companies, and we do a lot of market research with them just to, you know, learn about their research. They can obviously learn about our priorities and our current status. But yeah, I hope that you can see it if I type in our contact information into the chat box, and you can certainly email us there. So it's enact@hhs.gov.
Awesome. So that, again, for everyone on the line, is ENACT, ENACT, at hhs.gov. Awesome, and thanks for sharing that. Let's see here. So we have a great question coming in. Let's see, one from our good friend Lauren. Let's see, she typed it into the chat, but I think it is a really important one. So her question is, are all the tools being used in a single trial, or are they being used across multiple trials? And if so, has enrollment already started? And then the third part of that question is, what are the implementation or patient engagement challenges? Great question, Lauren. These are some of the ones that I struggled myself. Great questions.
Yeah, thank you, Lauren, for that question. So I'll try not to forget any of the pieces. So the first question is, are all of the devices joined in multiple clinical trials or just single ones? So due to the type of funding vehicles that we use, we are at this point only capable of running small clinical studies. And that's not just a matter of finance, that's also a matter of the mission of the entire division. So we are attempting to fill this valley of death space. And so I would think of the goal of our programs as kind of conducting pilot studies, basically seeing, how well a technology that's developed in the lab and maybe has been used in animal experiments, if that's appropriate, how well does it really fare in the real world, right? Do we really get, reliable results, et cetera? And so, for that reason, and then we've actually currently had smaller clinical studies for each project where a single device was used for the most part. We did partner with DARPA actually on a larger clinical study at different sites where we've used multiple different devices, but I think actually that they were assigned to different cohort members as well. So one way to compare all of this data, right, is to conduct a multiparametric study, right? And that's something that we're actually planning to do very shortly, is analyzing the clinical data from these very disparate clinical studies with very different sensors that measure very different things and make some sense out of it. And I think it's a difficult problem, but it's not an unsurmountable problem. And so, we've seen some of the wearables, clearly some of them also already have FDA approvals. Those clinical studies have, for the most part, been completed, though not yet analyzed. And then I've also presented a couple of technologies that are still really early on, where we're still working on the sensor development. And so those have not yet been tested in the field, if you will. And there was a third part of the question, I believe, but.
Yeah, sorry, you did a great job. Sure, yeah, great job answering those. So the third part is the third part of the question is it's a two-part. So what are the implementation challenges, and then also the patient engagement challenges, which is when we continuously ask ourselves in this space.
Yeah. So I think generally a lot of people are excited and wanting to help, but clearly compliance is an issue, and I think that many studies will probably deal with some kind of challenge when it comes to compliance. I think one of the issues is that you can't really have 3 or even 2 wrist wearables that are worn by one and the same person every single day for 2 months, right? I mean, people just don't want to have bulky things on their wrists. Maybe they'll wear one device, but not really 2. And so, comparing, kind of having a comparative study with 2 wrist wearables is a bit challenging. And that's, one of the many reasons why we're exploring all of these different form factors, right? And so I think, kind of the smaller the device, the less obtrusive it is, the less you have to think about it, I think is generally better. And, one of the things that we've learned also as part of this is that, battery life of our devices really matters a lot. And data storage capacity matters a lot. So it makes a difference to the study cohort if they have to do something every single day or potentially twice a day, if they have to charge the device, et cetera, or if they just have to do it once, right? And so we're always pushing for longer battery lives and greater data storage capacities to kind of make it easier on the subject. I would also argue that, and this is not necessarily tied to our current clinical studies, but I would also argue that, for these types of applications, choosing a cohort that has kind of a very personal investment in the study may actually help us get better compliance. For example, if you have a cohort that actually has a medical issue, let's say COPD, right? They have a personal investment in wanting to be healthy, in wanting to understand what's going on with them, even if the clinical study doesn't necessarily provide the data, right? So I think they may be a little bit more motivated than people who are just completely healthy and who just don't have this kind of personal investment, I would say. And then the third issue is, we've had a couple of just longitudinal studies. And we're looking, as terrible as it is for an individual to even get the flu and then be ill with the flu, we actually need those numbers to understand whether our devices and our algorithms can detect the signals. And, if you have a relatively small cohort and maybe 5 people actually get the flu, that's just not enough data, or not enough. It's not going to give you good enough statistics to be able to make a very definitive claim. And, that's why I think, a study, and I would, again, depending on the problem, I'd have to work out the statistics. But that's why really bigger studies, I would say at least, 100 plus subjects are really important. But that also means that, out of 100 subjects, 10 subjects with the flu is probably not going to be enough. We still need more. And so, that's obviously a challenge. And that's something that can be somewhat addressed by joining a challenge study, but we don't always have the option. And so, it is kind of a trade-off. And again, our way of trying to kind of circumvent this challenge, if you will, is having a lot of these smaller clinical studies and then conducting a multiparametric analytics approach where we put all of the data together and try to make some sense of it. I do hope that addresses your question.
Yeah, I think you did great, Shayla. Those are all kind of questions that every single one of us has to grapple with every time we do a trial. And so the things that you're saying is helpful for us. And we're all trying to work it out together. So I appreciate your insight there. So we're going to go to our first live question. So let's see, looks like Jen Goldsack, Dime's executive director, has got a question for you. Jen, would you like to go ahead and ask your question?
Yes, it's so fun to listen in, Shayla, and learn. Thank you so much for a terrific presentation. And Lauren, your questions were really good, and I was scribbling some notes here too. Shayla, I wanted to dig in a little bit to what you were talking about in terms of identifying health signatures. That makes a lot of sense. And I think your answer to Lauren's question as well started to tease out the need for sufficient data to really understand these signals. One of the things that I talk to folks a lot about is as we develop these sort of novel measures of health, or even as we measure very frequently aspects of health that we may only sort of capture periodically in the clinic currently. So things like it might be heart rate, it might be blood pressure. We need to establish normative data sets for these new measures and for the continuous or frequent measurement of even existing sort of health concepts. As you guys are sort of building these data sets to try and identify health signatures, how are you thinking about really establishing a deep understanding for today and the future of what normal data is for some of these measures, what you might expect from a healthy population? And have you had the chance yet to give any thought to how that might become a public resource? Because I know it's really important to the field.
Yeah. Thank you, Jen. So you were breaking up there a little bit, or it might be my phone, but I think I got the gist of your question, which is, how do we understand the normative data for patients, and if there's a way to create some kind of library that's publicly accessible. I believe that was the gist of your question. So I think what you're talking about here is really important. We need to understand, again, what the normal physiological range is, I will say, or biomarker range is for an individual. We are tackling that through different approaches. One of them is, again, just monitoring an individual for a very long time, even in a challenge study before they're actually exposed to a pathogen. And, we've, for example, learned that observing an individual just for 2 days before they're given, before they're exposed to the influenza, for example, in a challenge study, that's not enough. You cannot really establish a kind of a healthy signature or the normal signature for an individual over such a short amount of time. And, the idea, I think, overall, specifically when it comes to presymptomatic detection of infection is that these would be devices, wearables that a generally healthy person or really anybody would be continuously wearing, and the device would continuously be analyzing the data to establish some kind of baseline. So we certainly are thinking about it and are tackling it as well. I think that, you know, the component of making the data available and really kind of delving into it in a way that you're proposing, I think that's really important. But I'm not at this point quite sure that it necessarily fits with the mission of BARDA. I mean, it seems to me that this kind of exercise would be incredibly useful just to enable general research. And I, you know, think somebody should do it. But, you know, again, my division specifically, we're all about innovation and not basic research. And so even though we are attempting to address kind of a research question, which is, you know, can you detect something, an illness presymptomatically or not? I think that, you know, kind of the scope of gathering all of the data, and frankly, to do it properly, we'd have to not only look at different, you know, general population group, like by biological sex or by age or by race and ethnicity. We'd also really have to look at the genetic makeup, right? And certain variants and, you know, the lifestyle to really understand, well, what's a normal signature for, let's say, a smoker, or a normal signature for somebody with diabetes, et cetera. And so, you know, I do hope that we'll be able to do it in the future, but at this point, it's a little bit outside of our mission. So does that answer your question?
It does. It answers it very well. Thank you, Shayla.
Shayla, I've got some more questions for you. So it looks like you guys are doing a lot of projects, and some of them are rather early on, and some of them are, you know, perhaps going to even become bigger later. But whenever you ENACT gets big findings, how is it that your team is planning on sharing this information? Is there going to be maybe any publications, or is that going to be something that the other team works with? What's your plan for that?
Yeah. So, you know, I would consider these findings to really have two different natures. You know, there's obviously the work that's done by each of our partners. You know, they do their own data analysis and they come up with certain findings. And, you know, these are not grants, right? We operate with contracts. And so they are not required to publish their data in research journal, and it's proprietary information that they can choose to disperse in any way they wish, right? But that being said, obviously we work very closely with all of our partners, and we want to, you know, share their successes. And so we're actually, if you pay attention over time, we're retooling our entire website to, you know, much more closely reflect the successes and the advantages of our different technologies. You know, I've mentioned one of our partner companies has recently been very widely covered in the media. I think that's Sonica, and you know, that's certainly one outlet. They're very proactive about sharing their information about their technology, and you know, we obviously want to support them in that. So I would say keep an eye out on our website. Certainly, you know, we support our partners publishing white papers. Publishing data on their own websites, and especially I would say our academic partners have an interest, of course, in publishing research papers. And so, you know, we can also put, if anybody's interested in their work, you know, we can attempt to make a connection between an interested party and the company. You know, when it comes to I would say that data analytics project, we'll have to see how that's, you know, if the results are positive, we'll have to see how that's best dispersed. But I would say at first, you know, just a look at our website would be probably the best first place to go. And you can also see, I think BARDA in general has been quite active on social media. You can look us up on LinkedIn. I think we have a Twitter. We have several Twitter accounts. I believe Facebook as well. So I do suggest that you know, people do check us out on the social media, because that's also a really good way to find out what's going on with us.
Great, thanks for clarifying. I appreciate your response. So it's very clear that you know, even though ENACT and BARDA is, of course, federally funded, it's completely different fundamentally than, say, the NIH. So completely different expectations there. So thanks for clarifying that. I appreciate it.
Yeah.
I am looking forward to keeping an eye on BARDA and on your work to see really great stuff come out. And this is a really exciting time, and I think it's only going to get even more exciting and even better. We have another great question here into our chat panel here. Let's see. Looks like we got a great question from Nikunj Patel. So what are this is a great one. What are the do's and don'ts for a potential group considering BARDA partnership application? Great.
Yeah, thank you for the question. That really is a great question. So I would say absolutely a do is contact us first before you submit an application. Absolutely, you can submit an application, you know, as long as the announcement is open and accessible. But I strongly recommend that you talk to the relevant program manager, and I would argue that really holds for my entire division, if not broader than that. It's a really good idea because we can help explain what exactly we are looking for in a particular announcement, funding announcement. And, you know, we can kind of help, if you will, translate the language from our easy BAA, from our public announcement into, you know, what really needs to be in there. And, you know, we also obviously want to hear about the proposed technology, and sometimes we can get enough information to, you know, if it really doesn't fit with BARDA, we can suggest another funding source. Or we can say, yes, you know, it looks like your application could potentially be responsive to our AOI, please do apply. And I think sometimes people have a bit of a hard time actually finding our funding announcements. And so if you want to touch base with a program manager first, we can certainly help guide you through the entire application process. I think that saves, you know, everybody a little bit of time. And, you know, last not least, I think it's just generally a good idea to kind of develop a good professional relationship with any program manager. So, you know, don't is, I would say also, just read the funding announcement very, very, very closely. So you don't want to just submit anything that you think is a good idea if it doesn't fit an announcement, because what then happens is that if you're not responsive to the requirement of the funding call, then your application may have to be rejected, right? So we're looking at responsiveness first. And so, you know, I would hate to see somebody, you know, spend a lot of time writing a proposal and then having to say, but wait, this particular funding announcement isn't even accepting applications at a particular time, right? So, you know, again, talk to the program manager well before you are ready to submit an application, and then read our funding announcements very, very, very closely.
Great, thanks for that. It's always really good to get some more operational insight into how to get involved with ENACT. Really exciting work. Another question that I have that is closely related to that one is that I'm sure you have seen lots of really fun applications. And so I understand that perhaps the kinds of things that that ENACT is looking at is primarily early signal detection and wearables and really great things that are related to the theme of the products that you just shared. But my question is, what are kinds of what technology aspects are you most excited to see come across your desk in an application? What are the kinds of really exciting innovation types that you like to see, or maybe what are some that you maybe even see more often that ends up being outside of the realm of ENACT?
So what I meant to address earlier when I was showing these the pie charts that look at the different sensing mechanisms. I think the reason why I highlighted that is because I think it showcases that ENACT is really interested in very different approaches. So, and innovation in the sensing technologies. And I will take that even further. So I'm not going to say I'm looking for a particular thing or that thing excites me, because, you know, that might really actually limit people. Instead, I would say that I'd encourage people generally to really think outside of the box, right? Think about, let's say, and I've shown you various form factors, right? I've shown you, everything from a wrist wearable to an arm patch to essentially a Band-Aid that's worn on your clavicles, right? So can people go beyond that, right? What is a form factor that really works for people and yet gathers, the data? And, without giving specific examples, because again, I don't really want to shoehorn people in one direction, I would really urge people to think more about the various form factors and innovation in that space. And I think in that context, it's also really important to think about, the usability and the accessibility of these technologies, right? So, and I'll use the following examples. I see a lot of technologies out there that require the use of a smartphone. Well, we may think that a smartphone is really ubiquitous and that everybody has them, but truth is not everybody does, right? Or not everybody has, a sufficiently high, data speed to really be able to use it in this particular context. And so, I'd really like to encourage people to think about what is a technology, or what's a form factor, or what's a sensing mechanism that is truly, accessible to everybody in terms of cost, in terms of use, in terms of, can you get it at a CVS, or do you need to, go to a specialty store to obtain it? What's the ease of use? Do I need to, do really hard work to charge it, or to get data off of it? Or does the device somehow magically do all of this for me? So, kind of thinking in that direction, like what are some really novel form factors and how can we make any of these sensing technologies really widely available, so that it truly are accessible to all. So I'd certainly highlight this. And then, the other thing that I'd highlight is I presented 3 pillars to you, right, today. So it's, act, control, and treat, right? So it's information that can be best used by an individual or by a doctor or by an organization to make decisions. And most of these technologies were focused on, sensing devices, right? So I'd like to kind of push the boundary a little bit further and really, have people think about in which other way can they help provide early and actionable health information to anybody who uses it, right? I mean, for example, is there, technology that could be useful to, let's say, first responders? Imagine, for example, a mass casualty event, right? How do you figure out which patient with which type of injury should go to which hospital or which emergency room where appropriate resources are available, right? And so, that's less of a sensing mechanism, that's more kind of a data analytics problem, right? So I'd really encourage the attendees today to, not just think about a sensor that measures what's going on inside the body, but really about the broader term early and actionable health notification.
Excellent. Thanks so much for that, Shayla. Early and actionable health notification, and also equitable too. You always got to think about who can access it and does it help the most amount of people, not just the easiest amount of people. All right, Shayla Selamovic, thank you so much for your excellent presentation. You really were under the gun there. We had a lot of questions for you, but we really appreciate your excellent insight and you putting together this really fantastic presentation. So thanks again for that. And the slides on the recording will be. Yeah, of course. And we'll have you back too whenever you want to give any more information too about ENACT. So the slides and the recording will be available afterwards to really digest all this information. And I'd like to talk about something that DIME has really exciting up our sleeve, which is the playbook. I'm not sure if you've heard of this just yet, but our launch event is next week, September 16th, where this is the comprehensive essential industry guide for developing and deploying digital clinical measures. This is it. If you want one piece to help guide you through this crazy world of digital medicine, it is this playbook, which you can see that we've built it with really excellent partners and dear friends of DIME. So be sure to register for the event that we have next week. And while you're registering for our events, do keep in mind that we have we have the upcoming I went to the registration site. Well, that's good to know that it works. And we also have an upcoming DIME Journal Club. So this one occurs September 22nd, and it's featuring Elena Ismailova and our friend Ben, and I should learn his last name, and this is completely my fault. So it's fit for purpose biometric monitoring tool technologies, or biomet. And this is really exciting because whenever DIME first started, we put together some research teams. This is one of the first readouts from our research teams. And it's great work by some of our greatest partners. And so be sure to join the Journal Club to learn more. And at that, we are at the hour. So thank you everyone so much for joining us today. Thanks.